Bismillahirrahmanirrahim, and we start another paper 1-2, but this is an A-level biology paper, and this is the latest March 2021 paper which we're going to do today. Let's start with question 1. It says the photomicrograph shows onion cells, and it says the actual length of the onion cell is 350, labeled Y is 350 micrometers. So now if you do a simple calculation, you measure this whole length and say this is 14 centimeter. So 14 centimeter is equal to 140 millimeter. Now this is equal to 140, and you put in 3 more zeros, then this becomes micrometer, and then you divide it by 350, and the answer is 400. So this is how you were supposed to calculate this and find out. So you measure the actual length, and then if you measured it in centimeter, then you convert it into millimeter, and then you convert it into micrometer, because this is 350 micrometer. If this was 350 nanometers, then of course you'd convert it to nanometers. So please understand how we would do this question 1. Then coming on to question number 2. Question 2 is which row correctly shows the presence of a cell structure in typical plant cells and in typical prokaryotic cells. So here we're talking of plant, and prokaryotic means bacteria. So it says which row shows the presence of a cell structure. Now both of them, of course, centrioles are not present in either plant or in prokaryotes, and the lysosomes are not present, and cellulose cell walls are only present in plant cells. So 70S ribosomes is the only one which is present in both. And if you could correlate that by a typical prokaryote, you meant bacteria, because some bacteria have slime capsules. All bacteria do not have slime capsules. Some bacteria have plasmids. All bacteria do not have plasmids. Some bacteria have flagellum. Question 3. Which letter identifies cell structures where semi-conservative replication of DNA takes place? So semi-conservative replication of DNA is where is DNA present? Yes, it is present inside mitochondria. It is present inside chloroplasts. It is present inside nuclei. And eukaryotes have nuclei. So the answer was A, semi-conservative replication of DNA. I always tell you how to do this question. It's very interesting. You got to understand is if I make this circle yellow, right? Now let me make this circle red. And now let me make this circle black. Now you can see what does B mean, and what does A mean, and what does C or D mean, because you can see which colors are present and what does it mean by that. So when it says A is enclosed by yellow, red, and black. So this is how we've got to figure this out, how we had to find out which one was which. Then coming on to question number 4. Which parts of a cell contain ribosomes? Chloroplast, mitochondria, Golgi, cytoplasm. So only this one was the wrong one. Why? Because ribosomes will be present in chloroplast, yes, mitochondria, yes, and cytoplasm we have the free ribosomes. So 1, 2, and 4 only. Then coming on to question number 5. Which range of cell diameters is typical for prokaryotic cells? Now this is something that just you have to learn it. that what was possible. So 1 into 10 raised to the power 3 nanometers to 5 micrometers. So this is something which we have to learn and which we have to understand, is that it would be which are the dimensions of a typical prokaryotic cell. Question 6. Which row shows a combination of molecules that can be found together in a single isolated virus? The protein coat and a DNA strand. So that were the only things which were present in this line. The rest, there is no RNA, there is no cytoplasm, there is no phospholipid, there are no carbohydrates. Then coming on to question number 7, a student carried out 4 biochemical tests on a sample of food. Emulsion test was cloudy, Benedict's test was yellow, biuret test was purple, P protein present. Iodine was yellow. If it is blue-black, starch means present, there is no starch in it. Benedict's was yellow, emulsion was cloudy, so cloudy means fat present. Yellow means reducing sugar present. Which conclusion is supported by the results? Protein is present, yes. Starch is not present, the iodine was yellow. Glucose is present, fat is not present. 4 biochemical tests. So which conclusion is supported by the results? Fat is present, it was cloudy. Biuret test was purple. You see, we can't say glucose is present because Benedict's test is for reducing sugar. Reducing sugars are many. We don't know whether it was glucose or maybe some other reducing sugar, but we can't definitely say glucose is present. Many students would have marked that present because looking at the yellow part, because Benedict's, if it's yellow, means there is a reducing sugar. But which reducing sugar, we don't know, whether it's glucose or fructose or some other reducing sugar. There are many reducing sugars. So how can we say it's glucose? Now you're assuming this, that it's glucose. So this is, you see, you didn't read it properly. The only thing which was conclusive was biuret. Because if the biuret test is purple, it just means it's a protein, but doesn't mean it's amylase. Because amylase is an enzyme, that's also a protein. Pepsin is an enzyme, that's also a protein, but we can't say that it was supporting the fact that amylase was present, or pepsin was present, or insulin was present. Insulin is a hormone, and some hormones are proteins. So it only said protein is present, but it didn't have to, you can't sort of conclude that it was this protein, or it was a protein called pepsin, or it was a protein called amylase. So question number 8, which diagram represents a molecule of alpha glucose? You know alpha glucose, how you have to know the whole structure of glucose, and you know there's below carbon 1, the OH is below, but then of course you have to look at the other chemistry part as well, and that's why A was the answer. Now coming on to question number 9, which statements about glycosidic bonds are correct? Condensation between glucose and fructose uses water. Sorry, condensation is the formation of water. It cannot be using water, so this is wrong here. And why was 4 wrong? Amylose contains beta-1,6. No, just remember, beta is only in cellulose. Beta glucose is only in cellulose. That we know for a fact. So this was wrong as well. So that's why the answer is C, which is 2 and 3 only. Then coming on to question number 10. A triglyceride has dash bonds formed by joining fatty acids to dash. So has ester bonds. You see, remember this is glycerol. And to glycerol is attached 3 fatty acid chains. And this bond that is formed is called an ester bond. And this is called glycerol, not glycine. Glycine is an amino acid. So glycerol and an ester bond. This is something that is factual and we know this, so you need to be learning it. It's rote learning. Coming on to question number 11, the diagram shows a phospholipid molecule divided into three regions. X, it was here, Y and Z, this area, and R. So three regions, X, Y and Z, and then they talked about R. Now if you look at this, they told you that this was a phospholipid molecule divided into three regions. And then it says regions X, Y, and Z affect the properties of cell surface membrane in different ways. X, Y, and Z affect the property in different ways. Which row shows the effect of each region on the properties of a cell surface membrane? So increases permeability of the hydrophobic region, repels polar molecules, attracts water molecules. Now you see, what we had to understand, Y and Z was repels polar molecules, because Y and Z are the fatty acid chains. And these are the ones which do not allow water molecules to pass. So Y and Z was correct in this and this. So we had to narrow it down between B and D. Now what you had to look at is, was Y correct or Z correct? Now Z was an unsaturated fatty acid, while Y is a saturated fatty acid. And that was the discerning point. That's why the answer was D, because Z was the unsaturated fatty acid. You can see here. And question number 12. Which statement about collagen is correct? Tissues containing collagen fibers cannot stretch. Which statement about collagen is correct? Each polypeptide chain forms an alpha helix. Three helices are held together by hydrogen bonds in a collagen molecule. It says, what statement about collagen is correct? Three polypeptide chains form a single collagen fiber. No. They're saying which statement about collagen is correct. Which statement about this chair is correct? This chair is made of wood. So which statement about collagen is correct? You have to say to yourself, is this telling me about the collagen? Next, question number 13. Catechol is a chemical found in a number of fruits. Catechol can be oxidized to a quinone by the enzyme catechol oxidase. Catechol oxidase is inhibited by PHBA, which is structurally similar to catechol. In the presence of PHBA, the Michaelis-Menten constant Km for catechol oxidase increases. How does PHBA inhibit catechol oxidase? PHBA and catechol can both bind to the enzyme but not at the same time. PHBA is a non-competitive inhibitor of catechol oxidase. PHBA decreases the Vmax of the reaction. Here's the question, catechol oxidase is the enzyme. Catechol oxidase is the enzyme, right? And what is the substrate? The substrate is catechol. Right? Now it says catechol oxidase is inhibited by PHBA, which is structurally similar to catechol. So PHBA is something like this. Now that's a competitive inhibitor. So this one is wrong here. I know the answer is 1 only, but why is 2 wrong? Because you have to figure that out from the question. It said pH and then why was 3 wrong? PHBA decreases the Vmax of the reaction. No, competitive inhibitors do not decrease the Vmax of the reaction. When you increase the substrate, the Vmax is reached. So the answer was 1 only. PHBA decreases. If you could figure this out, that PHBA was a competitive inhibitor. This was what you had to figure out in the question, that PHBA is a competitive inhibitor. And you know, adding a competitive inhibitor does not alter the Vmax. So this is how you have to figure out the answer to this question number 13. 14. Which statement about the fluid mosaic model of a membrane are correct? Which statement about the fluid mosaic model of a membrane are correct? The movement of phospholipids in the membrane is reduced if the fatty acid tails are saturated. Glycoproteins in the outer layer of the membrane can move. Channel proteins are fixed in position. Fluid mosaic model means what? We have these phospholipid bilayer, right? And this is a cell. And this entire phospholipid bilayer is surrounding this cell. And what does it mean, fluid mosaic? Fluid means that these phospholipid molecules can move laterally. Fluid, just like water can move in a glass. So fluid. And what are the proteins? The proteins are present randomly. There's arrangement of proteins all around. There could be channel proteins, there could be enzymes, there could be anything, active transport proteins, carrier proteins. So the fact that the fluid mosaic model, it says which statements are correct. So the answer was 1 and 2. Channel proteins are fixed in position. No, I mean the whole thing moves, it's fluid. So how can they be fixed in position? The phospholipid bilayer is lateral movement of the phospholipid molecule. So of course if the proteins are present, they will also move. So that was question 14. Question 15, diagram 1 and diagram 2 show how the transverse section through a leaf changes when the leaf is moved from solution X to solution Y. Which row describes the water potential of the leaf cells and surrounding solutions in diagram 2 compared with diagram 1? Now you have to understand it. What has happened to it? The leaf was sort of curled. Now the leaf is straight. So what could have happened? Water potential of the leaf cells in diagram 2 compared with water potential of the leaf cells in diagram 1. It has become less negative because water has entered it. More negative means minus 100, less negative means minus 50. Now of course more water is entered, so it's become less negative. And water potential of solution Y compared with water solution X. Solution Y has lost water, so that has also become less negative. Solution Y compared to solution X. In solution X, what was happening? Water from inside the leaf was moving out. Now water from this is the water that was becoming dilute. Now it has become, the water has moved out, so it has become less negative. Yeah, I put some concentrations on it, that the X was 3% and Y was 1%. Now here you have to understand is water potential of leaf cells in diagram 2 compared with water potential of leaf cells in diagram 1. But then what have they asked you? Water potential of solution Y. Water potential of solution Y compared with water potential of solution X. So please read the question and understand the question before you figure out why is it both less negative. They compared Y with X. While they compared in this one of leaf cells in diagram 2 compared with water potential of diagram with 1. So you have to see how the leaf cells change and how the solution Y was as compared to solution X. So this was something which you had to understand. Then coming on to question number 16. Which features must always be present for water to move between two solutions by osmosis? carrier protein, cell surface membrane, selectively permeable membrane, water potential gradient. You see, which features must always two solutions by osmosis. So there has to be a selectively permeable membrane. We do this even in dialysis tubing. That isn't a cell membrane, that is a partially permeable or selectively permeable membrane. So selectively permeable membrane must always be present and there must be a water potential gradient. Yes, there in some cases we have aquaporins, but not always. So 3 and 4 was correct. That is why 3 and 4 was correct, because cell surface membrane need not be present like in a dialysis machine. There's no cell surface membrane, but osmosis is taking place. Even when we are doing a whisking tubing in the lab, an experiment on that, we can study osmosis. Question 17. When living pancreatic cells were placed in a solution of a red stain called neutral red, the cytoplasm became red. The cells were then removed from the solution of neutral red. The red stain in the cytoplasm moved into vesicles which were exported from the cell, eventually leaving the cell colorless. Which transport mechanism could explain how the red stain entered and left the cells? Entered and left the cells. So it says living pancreatic cells were placed in a solution of red stain. So we have a red stain, and what do? We put living pancreatic cells in it. And they say the red stain in the cytoplasm moved into vesicles which were exported from the cell, eventually leaving the cell colorless. So something was inside, formed a vesicle, so it had to be diffusion and exocytosis. It had to be diffusion and exocytosis. Exocytosis because it had gone out, and there was one was osmosis and one was exocytosis. The answer had to have exocytosis. Osmosis was incorrect. Dye doesn't move in by osmosis. Water moves in by osmosis. Dye moves in by diffusion. The red stain couldn't have moved in by osmosis. Question 18. Simple DNA molecules in a eukaryotic cell must be packaged compactly because their combined length is much greater than the diameter of the nucleus. Just like you have a reel of thread. If you've ever used a sewing machine and you'd put that reel of thread on the top, well, that thread is pretty long, it's yards and yards long, but then of course it's all wound around a spool. Which structures are required during the process of packaging DNA molecules compactly? Histone proteins. The rest of it was all rubbish. Question 19. Which statements about mitosis are correct? Mitosis results in genetically identical cells. Mitosis is involved in asexual reproduction. Mitosis occurs immediately after cytokinesis. No, that's wrong. Cytokinesis occurs after mitosis. Mitosis can be used to repair damaged cells. I keep on teaching you that. I said repair damaged tissues, not a cell. A cell cannot be repaired. It's impossible to repair a cell. You can repair tissues like you had an appendectomy so that you have a skin is cut, muscles are cut, but then the doctor stitches them together, and that repair is damaged tissue, not cells. I always tell this to you when I'm teaching you this chapter. So it was 1 and 2 only. Mitosis results in genetically identical cells involved in asexual reproduction. These two were wrong. Then coming to the last question, 20. A human chromosome is made up of different parts. Some parts of a typical human chromosome are more numerous than others. Which parts are listed in order from least numerous to most numerous in a human white blood cell? Centromere is which part? Centromere is that part on a chromosome where it aligns with another sister chromatid. So these are the centromeres. Then chromatids, these are sister chromatids. And then histone proteins. And histone proteins are those little balls around which the DNA is wrapped around. So it was simple to understand. It is listed in order from least numerous. So this side was the least numerous. And we knew that is histone proteins will be the most numerous. And there was only one in which histone protein, so the answer was A. Then question number 21. A piece of double-stranded DNA containing 12 into 10 raised to the power 3 nucleotides is transcribed and translated to produce a single polypeptide. What is the maximum number of amino acids in this polypeptide? So, as everybody knows, is that 3 nucleotides code for 1 amino acid. 3 nucleotides. So a piece of, but it's a double-stranded DNA. So that means on one side we have how many? And on the other side we have, so AT. The double-stranded was what? Was 12 into 10 raised to the power 3. So that means 6,000 this side, 6,000 this side. And then if you have 6,000 here, so 6,000 divided by 3, and this is 2,000. Right? It says 12 into 10 raised to the power 3. So you have something on this side and then 6,000 on this side and 6,000 on this side. So AAT is one codes for one amino acids. CCG codes for second amino acid. So 6,000 on one side, 6,000 on the other side, 6,000 divided by 3 gives you 2,000, and this was the answer. So please try to understand it. Why a piece of double-stranded DNA containing 12 into 10 raised to the power nucleotides is transcribed and translated to produce a single polypeptide or a protein. What is the maximum number of amino acids? So please understand this was a difficult question, but I'm sure you could have figured it out. The remaining part of this paper will continue in the next video, and thank you very much.